Monarch butterflies have become an increasingly hot topic in recent decades, and especially in the last couple of years. Conversations about monarchs often revolve around topics like their migrations, the importance of milkweeds, or other flowers that we can plant for monarchs. Rarely do the topics of monarch health or monarch diseases come up, which makes sense because most of us don't spend a lot of time thinking about insect diseases or health. Yet, these can also be fascinating and important topics to learn about and to discuss. So that's what we're going to do in this episode of the Backyard Ecology podcast, where we will be talking about a protozoan that parasitizes monarch butterflies. Nature isn't just out there in some far-off exotic location. It's all around us, including right outside our doors. Hi, my name is Shannon Trimboli, and I am the host of Backyard Ecology. I invite you to join us as we ignite our curiosity and natural wonder, explore our yards and communities, and improve our local pollinator and wildlife habitat. Hi, everyone. Before we get started, I wanted to thank all of my supporters on Patreon. Their monthly donations help make Backyard Ecology possible. If you would like to join them, please visit my Patreon page. I'll have links in the show notes for the Backyard Ecology Patreon page, blog, YouTube channel, and email list. Today we're joined by Dr. Sonia Altizer and Kade Donaldson. Sonia is a professor of ecology at the University of Georgia, and Kade is the project manager for Monarch Health, which is a community science project based out of the University of Georgia. Hi, Sonia and Kade. Welcome to Backyard Ecology. Thank you for talking with us today.
Thank you for inviting us to speak with you.
Yeah, we really appreciate the opportunity.
Oh, you're welcome. And I'm really excited to be having this conversation with you because monarch butterflies are something that so many people are interested in, but we don't always think about their health or some of the parasites that they can have. So I think we're going to have some really fun conversations here. But before we get started, can each of you just tell us briefly how you got started studying monarchs and why you're wanting to study their health specifically?
Ooh, so I started studying monarchs about 25 years ago as a graduate student at the University of Georgia, and it was completely by a fortunate accident. So I had started graduate school and I was really interested in understanding the spread of infectious diseases in animal populations, and a professor in the Department of Ecology at the University of Minnesota studied monarch butterfly mating behavior and reproduction. And she lost her entire captive colony the first year I was a graduate student to this mysterious disease. And so she came into my office and she had these sort of deformed and oozing butterflies, and she put them on my desk and she said, "Sonia, you like diseases. Can you figure out what's killing these butterflies?" And so I hadn't intended to study monarchs at all, but it turned out that this was a fascinating problem for these butterflies. And when we started looking at patterns in the wild, it became even more interesting. And so I've been studying monarch butterflies ever since.
And my experience is a little different. So I first became interested in monarch butterflies, I believe in elementary school, when we did some studies on them and their impact in the ecosystem. They weren't super complex or groundbreaking studies, but it was enough to get my interest in the topic. So then when I came here in 2018 to the University of Georgia, it kind of was a perfect collision of worlds. Dr. Altizer came and spoke in my freshman seminar class, actually, and her lab just sounded like something that would be really, really interesting for me to become a part of, first as an undergraduate, and then I stayed along for the past year after I graduated last summer in 2021.
Very interesting. Yeah, I always love hearing the different stories for how each of us ended up taking the paths that we did, because they're always fun and interesting, I think.
Yeah, I definitely think that's true. I think almost everybody that I've spoken with in the Department of Ecology through my years and my experiences has said that where they are now in life, they definitely could not have possibly predicted a decade ago. So I think that's a really cool testament to kind of leaving your options and your doors open and going where the world takes you.
And I forgot that you had studied monarchs before you came to UGA. And Kade did his bachelor's degree in the College of Public Health at the University of Georgia. So it's a really neat example of merging interests in public health and infectious diseases with looking at what's happening in natural systems with infectious diseases.
Yes, I agree. Because, yeah, my background's in wildlife biology, and infectious diseases wasn't really something that I was necessarily interested in or really thought about much at all as far as the disease spread and stuff with it. Occasionally they pop up in the wildlife populations, but it wasn't something that I really focused on. But more and more, I'm seeing how important that is really in having that study and having that crossover like you were talking about.
Yeah, and especially with butterflies. So people don't normally think about butterflies as getting sick. So, if we ask someone to name some infectious diseases, we probably could all easily name, 5 or 10 examples of human infectious diseases, and maybe some that infect livestock or pets. But we don't think about butterflies and other sort of non-pests, non-economically important insects as also having problems with pathogens and disease. And so it's really interesting to think about how the processes that might be affecting disease spread and the health of these butterfly populations are not too different from things that might affect disease spread and the health of people.
Yes, exactly. So I know one of the ones that you guys have been looking at a lot with monarchs is OE, or that's the abbreviation for it. So let's start from the beginning. What is OE? And I'm not even going to try and pronounce it. That's why I'm just going to say OE and leave it to you guys.
So OE stands for Ophryocystis elektroscirrha. And this is a protozoan parasite that causes a debilitating disease in monarch butterflies. This parasite is a specialist on monarchs, so it is mainly transmitted from butterfly to caterpillar. It cannot infect people or other butterfly species. There are some closely related protozoa that do infect other butterflies and a couple of moth species. But in general, this is a very monarch-specific protozoan.
Is it native then? Since it specializes on monarchs and monarchs are native to the US or to North America?
As far as researchers know, Ophryocystis elektroscirrha, or OE, has been in monarch populations for as long as, potentially for as long as monarchs have been around. It infects all known monarch populations to date around the world, so it's incredibly widespread with monarchs. For existing samples, we've been able to recover parasite spores from pinned museum specimens going back a couple of centuries. And there are some other butterfly species in the same group as monarchs in the milkweed butterflies that have parasites that appear to be similar to OE. So this appears to be potentially an ancient parasite in monarchs, or a native parasite that probably has moved around the world with monarchs as they have colonized different parts of the world.
And was this what your advisor had in her monarch populations when she came to you and said, "What's this?"
It is. And so this parasite was first discovered and reported from monarchs and a closely related butterfly called the queen butterfly, which is in the same genus as monarchs, in a paper published in 1970. So even though this parasite's been in monarchs for a really long time, it wasn't discovered until 1970. And then almost nothing was known about it until the 1990s. And I think part of the reason why people started becoming more aware of this protozoan is because people started doing more research on monarchs and captive breeding these butterflies in the lab and for other purposes. And I think we know for livestock that, whenever we bring animals into close quarters and rear them in crowded conditions, we tend to see problems with infectious diseases. And I think that's what happened with these butterflies. We started to learn more about monarchs. Researchers were raising them in the lab. There was also sort of the start of the commercial growing of monarchs for the butterfly breeding industry. that took off in the mid to late 1990s. And that's when people started seeing large numbers of monarchs dying from this protozoan infection and realizing, oh, there's something happening to these butterflies that we need to study.
Yeah, because that was something else I was wondering about, because I never really heard about OE until relatively recently. And I was like, well, did I just miss it because I wasn't paying enough attention in those circles at that time? Or was it something that wasn't as prevalent? And if it changed, then why? But it sounds like it had a lot to do with that more breeding and also paying more attention to it.
Yes, and in natural populations in North America, OE is quite rare. So historically, less than 5% of monarchs in Eastern North America were infected by this protozoan. So that means we'd have to sample 100 or 200 monarchs just to find a couple infected ones. In more recent years, infection prevalence has gone up, and that's something that we think is really interesting. And one of the reasons why we're excited to study this in our lab at UGA and through Project Monarch Health. But historically, infections in the wild were very rare. And so it wouldn't have been something that people would have commonly seen a few decades ago.
So is this something that's active year-round?
So it's very climate dependent, I guess, if it's active year-round. So in areas that have colder temperatures, we do see that the spores can become dormant. That's one of the big natural environmental controls that have been able to kind of keep the parasite at bay in past years. So we're seeing that one component, I guess, is global warming. As temperatures are potentially rising around the world, we're also seeing a potential increase in the range that OE can survive and the year-round nature of it, since less harsh winters may be conducive to higher infection prevalence. But in regions, so we were just talking about the general prevalence in especially the eastern United States being extremely low, around 5% or less in past years. So we've seen in areas like southern Florida, where there has been evidence of established year-round breeding colonies of monarchs, because there is milkweed that can survive in the conditions there all year round, which means spores are deposited on leaves year round and can survive there. So from that aspect, then I would say that the spores kind of aren't being controlled by any natural mechanism. And another contributing factor would be we have tropical milkweed that's been popping up a lot more recently in North America, especially, which is a non-native species, since it's mostly from around the Caribbean and closer to the equator is where it finds its home. So an issue with that is it can be somewhat hardier than more native milkweed species, meaning that it needs some harsher temperatures to die back in the winter. So even in more northern areas, if the winter temperatures aren't getting as cold, the plants aren't dying back, and the natural control mechanism of spores falling from the leaves to the ground and no longer continuing to be on the plant leaves is essentially removed by the year-round presence potentially of this milkweed.
Yeah, that's a great point. And so the idea is that in areas where monarch breeding is seasonal at more northern latitudes, parasites, because the parasite has to be transmitted from adult butterflies to the eggs and caterpillars, requires monarch reproduction to spread. And so in areas where breeding is seasonal and monarchs are only reproducing in the summer, that limits parasite transmission to just the summer months. But as Kade said, in areas where monarch breeding seasons have been extended by warmer winters, and or where, because of human planting of non-native milkweed species, there's now food available for monarchs year-round when it wouldn't have been several decades ago. When monarchs can breed over an extended period of time, and especially year-round, that really ramps up parasite transmission.
So you're saying that it's transferred like from the adult to then the eggs and caterpillars, if I'm understanding correctly.
Yes.
So what happens with the migratory population and they're overwintering in Mexico? Does the spores just kind of stay on them? And then when they start breeding in the spring, that completes the transfer? Or how does that work?
There's definitely potential for that. I would say that something that we see a lot in the environment and the study of ecology in general is that migratory culling can be a really, really important control mechanism. So I would say that for some of the more heavily infected butterflies that may be not as physically capable as uninfected ones, that's a control mechanism that would prevent those spores from ever getting to Mexico in the first place, since those butterflies would die off. That's definitely, I would say, a major part of it.
Yeah, during the fall migration, a lot of infected butterflies don't successfully reach the overwintering colonies, but some do. And so in order for the parasite transmission to start up again the next spring, when monarchs fly back north, some of those infected butterflies, they have to reach the overwintering sites. They have to persist throughout the whole winter and then migrate back to the breeding grounds in the spring to start transmission. So it's amazing to me. First of all, it's amazing that these tiny butterflies that weigh a half a gram can migrate these long distances to begin with. And then it's amazing, even more amazing, that some of these infected butterflies do successfully migrate. So a lot of the infected butterflies don't survive the migration, but some of them do. And at the times that we've been able to work at the wintering colonies in Mexico or in coastal California, when we find these really heavily infected monarchs there, I just think, how in the world could they have survived that migration being infected? And then of course they'd have to come back again in the spring to be able to carry the parasites with them to start the cycle again.
Yeah, like you said, that's fascinating. But maybe for people who, like me, don't really know more than OE is bad. I know that much, but I'm sure there's others who are like me who are listening to this who don't know that much more. Can you tell us, what's it doing? Why is it bad? Give us a little bit more details.
Sure, definitely. So the actual spores, the things that we would look at under the microscope as samples, are sort of the dormant form of the protozoan parasite. So that's what the adults would be depositing on their eggs, potentially depositing on leaves. So then when the caterpillars are hatching out of their eggs and they eat their way out, then they're potentially eating all of those dormant spores that are on the outside of their egg case and the leaf around it. So then once they ingest those spores, they're able to burrow, I believe, into the walls of their gut. And then they reproduce inside of the immature monarch. So then it doesn't pose itself as a really obvious characteristical threat until the monarch actually undergoes pupation and then eclosion. So at pupation, you actually can tell whether they're infected or not, but there aren't any real physical or behavioral signs before that. But you may notice, if you look very closely, a somewhat discoloration of the pupa of the monarch. And then once it closes, we can really see the more symptomatic and the obvious signs of OE. So it causes a lot of, in heavily infected cases, I'll say, it can cause major deformations, especially to the wings. So the monarchs may have trouble emerging from their pupal case at all. They may emerge and fall immediately, and then their wings might be crumpled either from the physical fall. They aren't able to spread them as you might have seen and dry them out properly, or they may just have had a malformation genetically due to the way that OE works.
Yeah, so the adult butterflies, in addition to them having trouble eclosing or getting out of the pupal case and having these wing deformities, they often tend to be smaller in size. And we've also found that the adult butterflies that are infected, they live slightly shorter than healthy butterflies. They fly less well. So their flight performance in terms of speed, endurance, how much weight they lose per kilometer flown, all these metrics are worse for infected butterflies. And then we also know that during the summer, infected monarchs mate less often than healthy butterflies. So it's sort of like death by 1,000 cuts, right? Like they have all these cumulative effects from parasite infection. And then the other cool thing is that the adult butterflies are covered with millions of these dormant spores on the outside of their bodies, but the spores have to be eaten by a caterpillar to cause a new infection. So the parasite replicates internally in the caterpillar stage and in the chrysalis stage, but then the adult butterflies come out covered with millions of these dormant spores. So it's really interesting that it's on the outside of the bodies of the adult butterflies, but it's on the inside of the bodies of the caterpillars when we can't see it.
That is interesting. And what you just said makes sense with it affecting their size and the wing strength and their flight patterns as to why most of them die off before they hit the wintering grounds in Mexico during the migration as well. And I'm assuming when you're talking about the spores being on the body, the dormant spores being on the body, and then coming off on the leaves and stuff like that when they're on the plants and laying the eggs and everything, that's basically like dandruff. It just kind of falls off. They're not actually trying to rub it off or doing anything like that. It just kind of, like I said, falls off like dandruff.
I think that's a great metaphor because, yeah, the spores just sort of fall off the body of the butterflies when they land on plants, when they lay eggs, when they're trying to nectar on milkweed, and then the caterpillars inadvertently eat those spores. And we've seen eggs laid by infected female monarchs that are covered in over 100 protozoan spores. And so the first thing the caterpillar does when it hatches from its egg is it eats the eggshell. So we know that even ingesting a single protozoan spore can cause an infection in a butterfly. So you can imagine these caterpillars munching on their eggshells when they're covered with dozens or over 100 spores, they're definitely going to get infected.
Right. Pretty scary stuff there when you start thinking about it.
Yes, and it is really sad to, so if you have, for example, a backyard where you have monarchs laying eggs on your milkweed and you want to rear some of those monarchs and enjoy the beauty of the butterflies eclosing from the pupal case, or teachers in classrooms that might be raising butterflies for a classroom lesson. It's really sad when the butterflies do get infected and they can't get out of their pupal case and they get stuck or they fall to the bottom and their wings are all crumpled. So it's definitely, it's not, this protozoan is not something that people get excited about or that most people would get excited about. But on the flip side, it really is a fascinating organism. So we've been studying this protozoan for a couple of decades now in the lab, and there's still so much to learn about it. And people often ask us, will your research help eradicate this disease from monarch butterflies? And our answer is always, well, no, and that's not our goal, because we think this organism, we know it's been with monarchs for a long time. It's interesting in and of itself. It has this life cycle that's closely intertwined with the life cycle of the monarchs. And so our goal isn't to eradicate it, but what we really want to do is understand how it's transmitted, how it spreads, what's happening in the wild, what might we as people be doing to inadvertently increase transmission? And then can we make recommendations so that we don't help the parasite spread more than it would naturally, if you know what I mean?
Yes. And that makes perfect sense because, I mean, like you said, it's been around forever as far as we can tell. And it's so closely associated with monarchs. And it doesn't seem to have been a problem until just relatively recently, when we as humans started getting more involved and tinkering and doing more stuff.
Yeah, and so we've seen from our monitoring efforts that over the past decade or so, even in the eastern migratory monarch population where they have to migrate so far and the infected monarchs don't migrate as well, infection prevalence by this protozoan has tripled over time. And so that's definitely a bit of a warning signal that something's happening in recent years to increase the transmission of this debilitating infection. And so we want to understand, why is that? What's happening to cause infections to increase? And then is there something that we can do to slow that down?
So how are you studying OE?
I can talk a little bit about what we're doing in the field, and then Kade, maybe you want to talk about some of our lab experiments and Monarch Health. So we can study this protozoan in the field by going out with our butterfly nets and catching wild monarchs and looking at infection prevalence in the wild. And so we've done this for many years, trying to measure infections in the overwintering sites, compare infections between populations in distant parts of the world. And we can measure characteristics of butterflies that we catch in the field. So we can ask things like, for wild migrating monarchs, are infected butterflies smaller than uninfected butterflies? Do infected butterflies have fewer fat reserves to help fuel the migration than healthy monarchs? We can also look at the immune defenses of these wild butterflies to see how they might be protecting butterflies from infection. And we can also study this parasite in the laboratory and through collaborating with volunteers.
Definitely, and I think I have a lot to speak on regarding that. I'm sort of at the forefront of both our community science project, Project Monarch Health, as well as a lot of our lab experiments that we've done recently. But I can talk about our community science project first. So Project Monarch Health as a whole, our goal is to understand the natural prevalence of OE in the environment, especially across North America. And we do this by reaching out to volunteers to see who's interested, sending them kits, which are for sampling the actual monarchs, which include data sheets to record all the information needed, like the location of the monarchs, the stage they were collected at, the date they were sampled, everything we would need to know to be able to get kind of better statistical methods to understand. So maybe the protozoan parasite is worse in certain stages of monarchs or in certain geographical areas across the United States, as we've seen a lot of evidence of. But then the volunteers will take little, I would say, quarter-sized clear stickers and adhere them to the abdomen of the monarchs themselves to get an imprint of some of their scales and some potential spores that may be on them. So that would be the OE spores. And this doesn't harm the monarchs at all. They're fairly hardy species. So as long as we're doing it fairly gently, we aren't harming them in any way. And then we're able to have those samples sent back to us in the lab, and we look at them under the microscope to establish how infected that monarch individual would have been. So our scale that we use for that, as far as gauging infection, is that if there's 0 spores on the sample, that would be 0. A score of a 1 is for samples that have 1 to 10 spores. A score of 2 would be for samples that have 11 to 100 spores. And then a score of 3 is for any samples with over 100 spores. So those heavily infected 3 samples would be most of them that we would see with some physical deformities if you stumbled across them in the real world. So those are the ones that are really, really likely to be covered in spores and have a lot of deformities. But I will say, you can't always tell that a monarch is heavily infected just from looking at it. So there definitely are individuals that, whether they exhibit resistance to the parasite, or maybe there was some horizontal transmission, like they acquired a bunch of dormant spores from coming in contact with another adult, potentially. So it's definitely cool to be able to sample these individuals. They may all have different phenotypes, different physical characteristics, but then at the end of the day, be able to say, yes, this is infected, no, it's not infected, or maybe just it's a little bit infected. But over the years, we've gotten, I want to say at this point, over 85,000 samples since we began in 2007. So we have a lot of data to work with. And from time to time, different graduate students or partner organizations will sort of look through the data, get some information on things like infection prevalence or how prevalence has changed over time, which I think is something really important to look at going forward. See, is the problem getting worse? Is it getting better? And what factors might be influencing it that we can change to sort of help the situation? And I can talk about some experiments we have going on now too. So one of our graduate students, Caroline Akins, last fall, she did a really cool experiment that was looking at how different rearing conditions for the monarchs as caterpillars may affect their ability to orient in their southern migration. So some really cool results came out of that. And I think it sort of bolstered our knowledge that while it can be a good thing to rear monarchs, we definitely want to do it in moderation, because the way in which they're reared seems to potentially have an effect on their ability to migrate and orient. So her experiment took some monarchs that were wild monarchs that were reared outside, so wild captured migrants reared outside as caterpillars, wild reared lab monarchs, so ones that were genetically our lineage monarchs that we keep inside of our lab, those were reared in the same conditions outside as the wild ones. And then we did the same thing, but inside. So we took wild caught monarchs inside and lab monarchs inside. And we used some really cool sort of pop-up tents, I would say, and we're able to measure, first of all, which direction they naturally tried to go in when they were in these tents, as well as we released some of them and got some vanishing bearings. So as far as the eye could see with our binoculars, what direction they were going using a compass. And the results were I wouldn't say they were surprising, but it was definitely interesting in a way that we hadn't seen before. So they strongly supported that the monarchs that were genetically wild and were reared outside were best able to go in sort of that southwest direction, southwest that we would be looking for towards Mexico. And the other three groups, whether it be from genetics, from rearing conditions, we don't know which of those might be a stronger driver, but it clearly seems that for monarchs that aren't reared outside and are genetically wild, that they may have a lower ability to orient and migrate.
That's something that's really interesting, something I'd love to be able to dig in deeper, maybe. And maybe that's a full episode that we have a conversation just about some of the migrating work and what some of these different research projects are coming out is, because I know there's been quite a few that are starting to look at some of that.
Yeah, we get a lot of questions about rearing monarchs, partly because we know that this infection can transmit pretty easily under captive rearing situations. And so people are concerned about preventing disease when they're rearing monarchs, whether it's, in their, on their kitchen shelf or garage, or you're rearing them in larger numbers. And then, there's also this idea that will reared monarchs migrate as well as wild butterflies. And I think there's some interesting science coming out on that suggests that no, they don't migrate as well as wild butterflies. I mean, some of them do orient and will make it to Mexico, but it seems like there's a really complicated series of almost like all the stars have to align properly to for a butterfly to migrate successfully, and that we can disrupt that sometimes if we bring them indoors or rear them. So this past summer, we also did an experiment that looked at the transmission of infection and the development of the parasite under different temperature regimes. And so one question is, will a warmer world be a sicker world? And a lot of people are interested in that question with respect to human infectious diseases like malaria or West Nile virus. But we're also seeing that for some wildlife infectious diseases, that as temperatures go up, infection risks increase. And so for these butterflies, we know that as winters get milder, that changes their breeding behavior and makes it easier for them to breed over a longer period of time and increase transmission. But a different question is, how do hotter temperatures affect the development of this parasite inside the butterfly? So we know that there are more frequent intense heat events or heat waves that are happening. And so how do these heat waves affect the butterflies and the parasites? We wanted to answer that question. So we did an experiment this summer where we exposed butterflies and their parasites to intense heat for different periods of time, and wanted to look at how does the duration of exposure to extreme heat affect how sick a butterfly gets. And I don't know if we know the answer to that question yet, because we just finished the experiment a few weeks ago and we're still analyzing the data. So maybe in a couple of weeks, we'll know the answer to that question. But our prediction was that the parasites might not do very well under intense heat. But I don't know, do you know the answer to that question yet, looking at the data?
I don't know the direct answer from our data, but I will say that one of our grad students in the past did a similar experiment, It was at constant temperatures of 34 degrees Celsius, which would be one of those maybe heat wave type scenarios of extreme warming. And it was fairly conclusive that the parasite isn't able to survive at a constant high temperature. So we would hypothesize that this experiment is going to give a result of longer exposure at that high temperature might sort of inhibit the parasite development, but we can't say for sure at this moment in time.
Yeah, my brain's already going with more questions to ask based on that. So I think we're going to definitely have to have you guys back again at some point.
Okay. But because the butterflies are so easy to work with in captivity, we always joke that, I mean, it's not really, it's true. We always say that monarchs are the tanks of the butterfly world because they're durable. They mate in captivity and lay eggs readily in captivity. They're easy to raise in under captive conditions. And the parasites are pretty easy to work with too in captivity. So it opens up this whole opportunity for asking different questions using experiments in the lab to help understand this interaction.
And it is really cool because you're studying it is the interactions. So you've got both organisms and their own biology going on. One of the protozoan that causes OE or that is OE, and then the other one of the monarchs themselves. And then you've got that interaction between them. So you're basically studying three different things here.
Yep. And we're really interested in how the outcome of that interaction is responding to environmental stressors, especially that these butterflies might be experiencing in recent years, ranging from thermal stress to exposure to pesticides or other environmental conditions like drought. So that's something that we're able to learn in the lab. How does that work by just varying one factor at a time? Whereas in the wild, all these things are happening simultaneously.
And while that's really interesting to think about, it's nearly impossible to study everything at once. You've got to start one piece at a time. And then once you got a good handle on each piece individually, think about, okay, now how do we start combining and how do they add to each other if we combine things, or do they mitigate each other and start looking at other factors like that?
Exactly.
Well, you mentioned the community science project, the Project Monarch Health. Who can participate in it? Can you tell us a little bit more about that? Because I know a lot of my listeners enjoy doing community science type projects.
Oh, of course. I would say it's pretty open to anybody. Our kits are completely free of any charge, and we're more than happy to send them out. So to do that, you would just reach out to us at monarchhealth@gmail.com, which I believe is linked to this. And you just send us a request. Hey, I'm looking to get involved in this project. It sounds like something that's interesting to me. And all we need is your mailing address. And of course, we love to hear any more information about how you got interested or what your background is. But as a baseline measure, all we need is your address to get interested, and that's it.
And are you looking for people more from like one area or another? And what times of the year are you looking at? Because like for me, I'm in Kentucky right now, and we still got some milkweed that's up, some of the common milkweed and stuff that are still up and have vegetation on them, stuff, but it's not going to last too much longer. I know down Georgia, Florida, it's going to last a little bit longer. So what's the kind of time periods and locations that you're really looking for people?
I would say we aren't exactly picky as far as time periods or locations. I know there are definitely some that are more optimal than others for volunteers, like either during the northern or southern migration, there's definitely going to be a lot more monarchs going through, so it'll be easier then. But as far as just getting involved with us, we're happy to send kits whenever, as long as you're able to keep track of it yourself, I guess, until you may be sampling monarchs. But I will say I was kind of surprised to find out earlier. I was looking through our master list of data. We have a lot of data from Florida, a lot of data from Michigan, a lot from some more like West Coast states. But I feel like a lot of maybe the Midwest, we don't have a ton of data on. I know, for example, oh gosh, I don't remember what one state we had only received 150 samples total, like in all of our what is it, 15 years now of doing our sampling, which compared to the 85,000 samples that we have in total, you can see that there's definitely gaps in sort of our knowledge base dependent on where we're actually getting the samples from. But I guess I'll kind of wrap that up by saying we're more than happy to get volunteers from anywhere.
And what does collecting a sample entail?
So I would say it's definitely something that takes getting used to. We have a pretty, I would like to say, detailed instruction guide and a bunch of instructional videos on our website on how to do it. But usually it would just be sanitizing your hands. We definitely encourage wearing gloves and swapping them regularly, just because you're potentially handling infected individuals and you don't want to be artificially transferring spores in between them. But as far as the sampling itself, it consists of removing a little quarter-sized clear sticker from our sticker sheet that we send out, and then you would sort of fold it over onto the abdomen of the butterfly, the same way that I guess maybe you would put a Band-Aid on your finger. And then you kind of just gently rub against the abdomen. Like I said, they're fairly hardy. We call them the tanks of the butterfly world. So I think it takes a lot of getting used to it first to make sure that you're not going to squish them. But they can definitely take a lot more duress than you would think. But as far as that, it's just sticking the sticker to their abdomen and sticking it to one of the note cards we provide. You can usually fit, I would say, about 10 to 15 samples on each note card. And then filling out the data sheet, which would include the collection address, which we are more than happy to receive street addresses. It's definitely more accurate if you are able to get GPS coordinates, but definitely not required, as well as the date that each individual monarch was collected, the stage that it was collected at. So if you're doing this in the wild, catch and release, you would be collecting the adults and sampling them, because I don't know if I mentioned this, you can only sample the adults. That's what we're looking for, since they're the ones with the dormant spores on the outside of them. And then, so we just need collection date, stage, sampling date, and usually sexing male or female, which we definitely have guides for on our website if you aren't experienced with determining the sex of a monarch. And then as far as catch and release, so like I said, it doesn't harm the monarchs in any way. So you're more than welcome to sample a monarch from the wild and then release it again. But we do recommend that you take something like a permanent marker and just mark a little dot on one of the discal cells of their wings, so that you know if you catch that same monarch again, you already sampled it.
And yeah, that's one of the things I've always been a little bit nervous about, is trying to handle a butterfly. I mean, I've done bird banding. I work with little bitty warblers and stuff, and they've got some of the teeny tiniest legs there are, but I'm like terrified of trying to catch a butterfly and hold it. And I'm like, I'm just gonna squish the thing, and I don't want to do that. And then now you're talking about marking on the wing. That doesn't do anything.
Well, tiny mark. So yeah, we encourage people not to make a big splash with a Sharpie, but just like a teeny tiny mark.
Mm-hmm.
I mean, when you think about it, these butterflies, again, they're another really cool community science project focused on monarchs is Project Monarch Watch, where volunteers put little tiny stickers the size of a hole punch on the monarch's wings, and they can, again, successfully migrate with that little sticker on their wings. And so we just recommend making a tiny mark with a little Sharpie marker on the wing, just to be able to tell that monarch's been sampled before. But they're tough butterflies. We've obviously handled, I don't know how many thousands in the lab. So we're used to it, but we do have a couple videos on our website that can show volunteers how to handle a monarch safely, and that sort of just walk people through the sampling process so that volunteers can see the best way, the best technique. And a couple of other things that might be worth mentioning are that there are a wide range of ages and backgrounds for people that participate in Project Monarch Health. So one thing that blew me away when we first started this project was that there were a number of school teachers that would participate with their classes. And these weren't all middle school and high school students. Some of them were working with 3rd, 4th, and 5th grade students, where they would have the students do the sampling and even sometimes look at those samples under the microscope in their classrooms before they sent them to our labs. And that was so exciting to me that there were young kids that were getting exposed to this world of things that could potentially infect butterflies and could really understand the concepts behind the project as well as the methods. And so a lot of our volunteers are retirees, nature enthusiasts. We get a lot of sort of adults working with homeschooled children, nature centers, and so you don't need any special training to participate in this project. And another thing that I wanted to throw out there that I found very interesting, because you mentioned, are there any parts of North America or any places where we might be most excited to get more samples from? And one thing that puzzles me is that there's a bit of a hole in the middle of the United States where we don't have that many samples. And so states like Kansas, Oklahoma, Missouri, Iowa, Ohio, Arkansas, even Kentucky, we don't have a lot of samples from those locations. And I'm not sure why, because we know monarchs are there. So if any of your listeners are from those places and would be interested in participating, we would love to send you a sampling kit.
Definitely. And I will keep passing the word around too as I talk to more people about it. Try and fill in some of those holes. So what are some of the questions that you get most commonly from participants about who are wanting to participate in this?
Definitely. I would say probably our all-time number one question, I think we touched on a little bit earlier, is how one can be responsible about rearing monarchs. So if you're going to bring them inside, what proper practices you should follow to prevent maybe parasite transmission, or just making sure that they're best set up to survive in the world after that, which I will say we have guides for on our website, which is, like I said before, linked to this podcast. But I think if I just want to talk about it a little bit, we definitely encourage people to get that experience. I mean, it's a great way to be connected to nature, to see the life cycle in front of your eyes. But I will say it's definitely best done in moderation. We don't encourage people to rear hundreds and hundreds of monarchs for the purpose of trying to augment the population, because research has shown that that might not be an effective method and it might actually hurt them in the long run. But we think it's a great idea for people to get as involved with nature as possible and sort of get that experience, get some passion for the world around them.
Yeah, I think people really want to know what they can do to help monarchs, because we know monarch migration is in trouble. And so a lot of the questions that we receive from volunteers and monarch enthusiasts relate to what are some ways that we can best help monarchs, whether it's planting milkweeds in our yards. And so we have tried more and more in recent years to encourage the planting of native milkweeds. So encourage folks to find out what milkweeds would commonly occur in your respective region and figure out how do you access those plants or seeds and cultivate them, because there are over 100 different milkweed species in North America. There are so many cool milkweeds. Monarchs use over 24 different species of milkweeds as host plants. And so really trying to propagate those native milkweeds is something that we encourage. Nectar plants, we also encourage people to plant nectar plants for monarchs, especially in the spring and in the fall when they're undergoing that journey north and the journey south to overwintering seasons, because they need that nectar to fuel the migration. Another question that we get asked a lot is, what should I do with infected butterflies? So volunteers might find an infected monarch, so they might know just by looking at it that it's infected or it's sick, and they want to know what should they do with those butterflies. And that's a surprisingly complicated question to answer, because my answer is always, it depends. And so I think, and what we try to tell folks is that it depends on, first of all, how sick or debilitated the butterfly is. So it's obvious, if it's obviously deformed or struggling, we think the most humane thing to do with those butterflies is to put them in an envelope and put them in the freezer. So insects are cold-blooded organisms. They're ectothermic. As they're chilled down towards freezing, their metabolism slows down and shuts down, and they basically go to sleep. And so we think that the best thing to do for butterflies that are really visibly sick is to put them in the freezer, to euthanize them. But if they're not, visibly debilitated, then I think the question is even more complicated, because what I always tell people is my recommendation for what to do with those butterflies, whether to release them back into the wild or not, depends on whether a volunteer thinks that they themselves might have caused that butterfly to get infected. So if we sample the butterflies in the wild, flying around our backyard, and that butterfly is infected, and we didn't do anything to cause that infection, I personally think the best thing to do is just let the butterfly go, because it was it was infected to begin with. It was out flying around. But if there's a reason to think that a volunteer did something that might have caused the infection, for example, by raising the butterflies under crowded conditions where they were being exposed to potentially other contaminated monarchs, then it could be that the best thing to do is to euthanize those butterflies and not release them into the wild, because then that would be artificially augmenting infection.
And I'm assuming that when people are out there catching butterflies, especially if they're using a net and stuff, that sanitizing the net is going to be important too, because that could be another potential transfer mechanism. I mean, just like with the gloves that you guys were talking about earlier.
Absolutely. Now, we recommend that individuals who are out catching wild butterflies with nets maybe bring several nets with them. If they're catching more than a dozen butterflies a day, you have several nets and rotate them, and then sanitize them overnight to use fresh nets the next sampling interval. But because these butterflies are covered with millions of these dormant spores, and we know these protozoan spores can live a long time on surfaces, it's possible that contacts that infected monarchs make with things like nets, countertops, even people's hands, could be a vehicle for transmission of spores.
So how long can the spores live?
Well, I would definitely say it depends on the conditions. If they're in direct sunlight, a lot of UV radiation, then it might just be a few minutes, a few hours. So if you are out catching, you're rotating between nets, you're leaving them in direct sunlight, you might be okay. If it's something like, say, your kitchen refrigerator or in the soils, it can be a lot longer. When we are conducting experiments in which we're using these dormant spores to purposefully infect monarchs as a mechanism to study other variables, we've seen that spores from years past are still able to be used to recolonize the guts of these monarchs. So it's definitely something where you're a lot better safe than sorry. You definitely want to make sure you're thoroughly cleaning all of your stuff. Most of the time we recommend a 20% Clorox bleach to water solution for most harder surfaces. So our countertops in the lab, that's what we use for them. And then some nets you can get away with using that concentration, but I would be very careful about the amount of time. So for some nets, you might want to stick closer to maybe 5% or 10% for just maybe 15 to 30 minutes and rinse them off immediately afterwards, or else you're going to have your nets eaten through a little bit. But yeah, the spores, they're very, very hardy. Since they are in that dormant, non-active form, they can survive a lot of environmental stressors.
Yeah, it's amazing that the spores can live for many weeks to many years. And if they're protected from UV light, if they're protected from extreme heat, if they're protected from freeze-thaw cycles, they can stick around and remain viable for quite a long time.
Yeah, sounds like it. And what you just said kind of answered my next question, because as Kade was talking, I was like, well, as the milkweed dies back, if it's on the milkweed, then it falls on the ground for the winter. When the next spring, the new growth comes back up again, could the spores get on the milkweed there and just directly infect from the soil? But it sounds like with that freeze thaw cycle, like you were just saying, that's probably not a likely possibility.
Absolutely. In areas where there are harsh winters and there are repeated freeze thaw cycles, that appears to be able to effectively kill most of the protozoan spores.
It's just one of those things that popped into my head as we were talking. I was like, well, wait a minute. What about that? Especially in a place like Kentucky, where we don't know what our winter is going to be. It may be warm, it may be cold. Yeah, we never know. And it changes from one day to the next too.
Yeah, I think even as far south as Kentucky, Tennessee, Georgia, unless you're really in the extreme southern US or the southern coast, or on the west coast, like in coastal California. I think the winters in most parts of the US are not going to be suitable for this protozoan to successfully overwinter.
And that makes sense too, given how uncommon it seems to have been previously. Seems like it would have been more common back then if it could overwinter like that.
Yes, exactly. And I think that also leads into a related question, which is, what can people do to try to limit the spread of this parasite? And so one thing that we recommend is for people who live in areas where there are mild winters, where they do get green vegetation all winter long, we really recommend to try to keep resources seasonal for the monarchs. So to cut back the milkweeds if they have milkweeds that might be able to persist throughout the winter months, and to really plant those native milkweeds, because almost all milkweeds that are native to North America do naturally die back during the fall and winter months. And it's largely the milkweeds that come from the tropics that don't have that annual dieback period that would be able to provide food for monarchs all winter long. So we really recommend that people living in the United States, for example, do what we can to keep milkweed seasonal, to try to limit the extent to which the monarchs can breed year round. Because it's almost universally where we see monarchs breeding year round on milkweed plants, infections are going to be high. And by high, we mean 80 to 100% of those butterflies are going to be heavily infected with this protozoan, causing problems to the scale of seeing multiple deformities, just walking around looking at butterflies in the wild. And so that's something we don't want to encourage.
Right. And I'm sure the people who are planting the milkweeds don't want to encourage that either. They're doing it because they want to help, and they don't realize that they're not helping, they're actually harming. And so it's finding that way to gently help them understand without pointing a finger and saying you're being horrible and awful, but just as you learn more, you do better. As you learn better, you do better. That's the way we all are. So
yes,
yeah, I think that is very important there.
Exactly. And these tropical milkweeds that are the non-native milkweeds that can stay in leaf and flower all winter long, I mean, they're beautiful plants and they're super easy to grow. And I'm even guilty of having grown them in my garden from time to time. But just, with greater knowledge, understanding that, to the extent that it's possible to replace them with native milkweeds when those plants do become available, and to try to cut them back or just cover them up in the winter with screen or maybe tents so that they're not available to the butterflies to be able to use all winter long. There's just some simple steps that people can take to try to limit the transmission of this protozoan.
Yes.
One interesting thing, starting about 10 years ago or so, it was community scientists and volunteers that I think first opened researchers' eyes to this interesting winter breeding phenomenon in the southern US. So volunteers that submitted observations through a different community science project called Journey North was what first opened researchers' eyes to this idea that, hey, wait a minute, there are monarch butterflies staying in the US all winter long when most of the population is overwintering in Mexico. And so being able to communicate with these volunteers and find out, what are the monarchs doing in southern coastal Louisiana or South Atlantic Florida during the winter months? That there are milkweeds available for them in these places, that they are breeding during the winter months. That whole phenomenon first came to light through observations of volunteer citizen scientists. And then more recently, we were able to look at monarchs in these places to see that they were 5 or 10 times more likely to be infected with this protozoan than migratory monarchs. So these volunteers and these community scientists were just instrumental in these important discoveries about monarch migration ecology and about the transmission of this protozoan. And we would never have been able to answer some of these questions if it weren't for the observations of volunteers.
And that's just another example of how important it is for everybody to be looking and sharing what they're seeing. And I mean, that's one of the reasons I've always loved community science and citizen science, is because of that. And because there's always so much more to learn, and none of us can know everything. So we just learn from each other.
Yeah. And so when people ask, what can they do to help monarchs? Another answer is participate in one or more of these community science projects and share your observations with the broader community. Because through these projects, like Project Monarch Health, Project Monarch Watch, Journey North, broader projects like iNaturalists, volunteers and nature enthusiasts and scientists can see in real time what's happening at really big scales. So we can look across the whole continent to see, oh, what are the butterflies doing this week? And that is so powerful, to be able to share those observations and have them inform scientific knowledge.
Yes, I agree. Well, this has been really interesting, but I know we've been talking for about an hour and I could keep going. Definitely would love to have you guys back again sometime to talk about some of the other things that you guys are studying and learning. And there's just so many different ways we could go with this. Is there anything else you would like to share with us before we wrap this up?
I think one thing just to share is that monarch butterflies are so they're just one butterfly, right? They're one species. They're so visible, they're beautiful, they're charismatic. I think people form a connection with monarch butterflies oftentimes early in life, especially maybe as school kids in the US or other parts of North America. So these are special animals to a lot of people, and they have this amazing migration. They have this really important interaction with the plants that they feed on. And so there's so many unique things about these butterflies. And yet, again, they're just one species. And so I think to the extent that we can learn about monarchs and their migration and what they need to migrate successfully and take steps to help protect that migration, we're going to be protecting a lot of other species, a lot of other migratory species, other insect species along the way. So for me, one of the reasons why it's important to understand and protect monarchs is because they are part of these incredibly diverse communities and these food webs and these interactions with dozens or more other species that are important to protect.
Yes, I definitely agree with that. It's an ecosystem.
Mm-hmm.
We're using one species as kind of focal point, but we're really needing to create an ecosystem.
Yeah, and so part of our goal is to protect monarchs, but I think for us and for other people, the more important goal is to protect natural ecosystems. And so protecting monarchs, the habitats that they rely on, the other species that they interact with, even the predators and the parasites that attack them. These are all very important parts of the ecosystem. And so I try to tell people, it's important that you study the butterflies in your backyard. You tell us what's happening with them, but be aware that, they're feeding on plants that other insects might be eating too. And there might, even though monarchs are toxic to predators, there are things that eat them. Mm-hmm. And it's not that we just want to save the monarch butterflies in a bubble. We want to save the whole network of complicated interactions that they are a part of.
Yes, definitely agree with that. And I mean, monarchs produce a ton of eggs. I mean, all butterflies do, and most of them don't survive. And so it's okay if the predators eat some. That's what those extra ones are there for.
Yeah, I know it blew me away the first time I realized and learned that only 1 or 2 out of every 100 monarch eggs that are laid will ever survive to the adult stage. And that's how, for monarchs, that's how it's been. That's a part of their natural ecology. And a single female monarch can lay 500, 800, sometimes over 1,000 eggs during her lifetime. And so having just 1 out of 100 survive, that's enough. And so being aware that these are animals that they live under tough environmental conditions naturally, and that they have been able to do just fine, to thrive for many centuries or longer. Certainly there are things happening in today's world that are changing the ecology of monarchs and maybe moving them towards a new normal. We want to understand what that new normal is and how can monarchs persist in the future in that new normal. But at the same time, they do have this amazing ability to survive under tough conditions. It's so inspiring that your listeners have this understanding of, of complex nature, and that really understanding how species fit into a bigger food web and a bigger picture is important.
That's one of the things I believe, and I try and convey that and share that with others. But yes, thank you guys so much for joining us today. And I will definitely have links to Project Monarch Health and everything we've talked about in the show notes. So people can just go there and find out more information. But yeah, thank you guys so much for joining us, and have a great day.
Thank you so much for having us.
Yes, thank you for inviting us. It was a pleasure to speak with you.
You're welcome. And thank you guys so much. Bye-bye.
Bye.
I appreciate Sonia and Kade taking the time to talk with us today. I found the conversation very interesting and educational. Like I said during the episode, I have lots more questions I would like to ask and topics that I would like to explore with them. Prior to this conversation, I had heard of OE and its effects on monarchs, but I wasn't aware that it was a native parasite that apparently has pretty much always been around. It's interesting to think about and study what has changed in the last few decades that has made OE so much more prevalent than it historically has been, and how can we fix what we appear to have broken. I highly encourage anyone who lives in the U.S. and is comfortable handling monarchs, or is comfortable learning how to handle monarchs, to consider participating in Project Monarch Health. Let's help them fill in those holes in the data that they were talking about having, because having lots of people from all over the country submitting data is the only way we will be able to better understand the changing picture of monarch health and how we can best help these beloved butterflies. If you find value in the Backyard Ecology content, please consider making a donation. You can find out how at www.backyardecology.net/support. Until next week, I encourage you to take some time to enjoy the nature in your own yard and community.